Greetings, I'm Miguel Ángel Rodriguez, talking about the article Fourier Transform Temporal Diffusion Spectroscopy, published in the Journal of Magnetic Resonance. This study is the result of a collaboration between the Institute of Biomedical Engineering and ETH Zurich, the University of Zurich, as well as the University Medical Center Freiburg in Germany. Authored by Franciszek, Hennel, Hannes Dillinger, Jochen Leupold, and Klaas Pruessmann. The article explores an application of Fourier transform techniques to temporal diffusion spectroscopy. Fourier transform diffusion spectroscopy, or FTDS, is a modern technique that significantly advances the diffusion phenomenon in complex materials and biological tissues. Building on temporal diffusion spectroscopy, FTDS introduced Fourier transforms to address limitations of earlier methods, such as limited frequency resolution and poor sampling at low frequencies. The technique quantifies how nuclear magnetic resonance signals attenuates as molecules diffuse through a medium. Instead of evaluating this attenuation in the time domain, FTDS transforms this into the frequency domain. This shift reveals the contribution of different frequency components to molecular modeling, enable a more detailed understanding of these directions within internal structures like cell walls and membranes. In practice, FTDs use a broadband gradient waveform repeated over time with variable delays. These delays introduce harmonic modulations that decode the spectrum of diffusion of frequencies into the spin echo signal. A discrete Fourier transform reconstructs the pure diffusion spectrum from these modulated signals. Unlike oscillating gradient spin echo or OGSE methods, which probe one frequency at a time and are limited to low frequency due to relaxation constraints, FTDS captures a wide variety range simultaneously and with greater efficiency. This approach has proved especially effective for detecting restrictive diffusion, particularly in biological samples where cellular microstructures influence water mobility. In one experiment, a celery stalk was examined using both FTDs and OGSE methods. When diffusion gradients were applied perpendicular to the vascular bundles, FTDs revealed a pronounced dip at zero frequency in the collenchyma tissue, evidence of restricted motion deep the cellular bundles. In contrast, gradients aligned parallel to the bundles yield a flat spectrum, indicating unrestricted diffusion along the cell lengths. These spectral patterns indicate the tissue architecture and differentiate between the normal and pathological structures in biological imaging. Beyond experimental capabilities, FTDs improves data reliability. Intense spectral sampling and Fourier-based signal processing reduce noise and enhance resolution, allowing for detailed microstructural characterization. Additionally, signal averaging further suppress random fluctuations, contributing to clear interpretations. FTDS was implemented on a 400 MHz nuclear magnetic resonance spectrometer equipped with microimaging gradients. Custom pulse sequences were incorporated, precise timing chips while minimizing signal distortion. Measurements using both spin echo and simulated echo sequences result diffusion spectra up to 120 Hz. Compared to OGSE, FTDs consistently deliver a higher resolution spectra with more data points, while maintaining agreement in overall trends. This method is not limited to plant tissues or simple systems. Its flexible methodology is applicable to a wide range of biological and porous materials, offering a non-invasive approach to probing internal microstructure. Whether studying water transporting plants or evaluating tissue cellularity, detecting early response to cancer therapy, FTDs provides the spectral precision needed for meaningful insights.